This video is meant to supplement some scientific background as well as material background on the process of reverse transcriptase PCR for our Fundamentals of Biochemistry lab. Our first section lets us utilize reverse transcriptase PCR to analyze the expression profiles of desired messenger RNA. To begin, we have our model organism over here that we are using in lab, the zebrafish. So we'll be exposing these guys to different kinds of compounds for a specific length of time post-fertilization. We'll talk about this in lab. And when it comes time for us to analyze and quantify the messenger RNA for our genes of interest, it's helpful to review the process of RNA transcription very briefly. So, as we all know, DNA is stored in the nuclei of the cells of our zebrafish here. We're going to zoom in a little bit and focus. So here is our DNA strand. We have our template strand and the opposing strand here. So depending on what is going on in our zebrafish, whether a stimulus is applied or an insult occurs, something has happened to our zebrafish to activate genetic transcription of our gene of interest, right? So something has occurred on the outside of the cell, and now the cell is going to transcribe messenger RNA. So our RNA polymerase comes in here, it reads our template strand, it generates our opposing strand of RNA. Our poly A tail here is added by another enzyme, and I've mentioned this in class, but the poly A tail is included to help slow the degradation of our mRNA once it's been spliced into the correct sequence of exons and transported into the cytoplasm. So the poly A tail here, again, slows degradation of our mRNA once it's been processed and exported out of the nucleus. So here we have, up here, we have our nucleus again, our nuclear DNA. We have our cytoplasm, and if you notice here, we have our mRNA strand from the previous slide. So we have our mature mRNA floating around out here in our cytoplasm, and we have, if you look closely, these other forms of RNA. Right, so in the cytoplasm, there are additional types of RNA, such as tRNA, so transfer RNA. There's rRNA, such as ribosomal RNA, and there's also forms of interfering RNA, which work in a number of ways. We won't talk about too much in class, but these types of RNAs can actually silence other genes. Some of them can even activate proteins in the transmembrane. Right, so now we're going to pretend we are in class and we're about to perform our RNA extraction from our zebrafish. So they've been treated. We have them in lab. We're going to anesthetize them and euthanize them. Once we homogenize their tissue, all the tissue transfers into one of our centrifuge tubes. So at this point, we're going to pretend we've completed our RNA extraction. So now in our tubes we have just the RNA from our samples. And if you look closely, again, all these different types of RNA are now in our samples. So that includes our desired messenger RNA, our tRNA, the ribosomal RNA, all the different kinds of RNA, right? So our first step in purifying and analyzing our gene of interest is to select for our messenger RNA. So we do this by applying a primer set that's going to set up for our D oligos. So this is an artificial DNA sequence that's going to complement our poly A tail here, and this is going to link right up here and allow us to select for only our messenger RNA, right? Because our other types of RNA don't have our long poly A tail. So our oligo binds up here. We perform a quick cDNA synthesis, so it's a short chemical reaction using a thermocycler. And now in this tube, we've also added an enzyme called reverse transcriptase, and then plenty of other nucleotides and other resources that our reverse transcriptase here is going to need. And if you recall, reverse transcriptase is so special because it can actually build DNA from an RNA template strand, right? So now we've performed our cDNA reaction. We have the production of a cDNA complement to our mRNA of our gene of interest, and this has occurred in a 1-to-1 ratio. So if there was one strand of mRNA floating around in the cytoplasm, now we have one strand of our complementary DNA. In reality, there's hundreds or thousands of our mRNA floating around in our samples. So the important thing to remember is that this is a 1-to-1 copy process, right? And then we'll treat our samples with an alkali wash, which will get rid of our original mRNA. And then the only thing left in our samples is our complementary cDNA. And then this is gonna have a complementary strand also built and added to it when we move to our next step. So through this process, we are selectively creating DNA that's specific only to our mRNA, again, from that poly A tail. So the only thing we've amplified now is messenger RNA. We're leaving the rest of the RNA behind. All right, so now we're on to the step in reverse transcriptase PCR that is responsible for amplifying only our gene of interest, right? So previously in lab, we've designed primers that are specific for our genes of interest, and today we're going to see how those work. So we've gone from our single strand of cDNA to a double-stranded DNA. I've taken the liberty of just cutting out that process, but know that this process occurs with a normal, quote-unquote, normal double-stranded DNA segment. So again, the only DNA present in our samples now have been generated by copying, reverse transcribing, rather, all of our messenger RNA from our samples. We've left all the other forms of RNA behind. So the general process for how this works is that through the use of a thermocycler, which is a machine that will literally cycle through different temperatures over and over and however they are programmed, we'll heat up our samples, allow some chemical reactions that I'm going to describe in a second to occur, and then cool back down. So as our samples heat up, we actually break the hydrogen bonds that help contribute to keeping our complementary DNA strands together here, right? So we heat it up, the two DNA strands separate. That's going to allow the chemistry that I'm about to describe happen. And then once our samples cool back down, our DNA strands find each other again and group back up based on their complementary nucleotide sequences. So during the first step of our actual PCR, so polymerase chain reaction, we heat up our samples, our two DNA strands separate, and so before all this occurred, we loaded up our sample tubes with a bunch of different nucleotides, a special DNA polymerase that can tolerate and function in high temperatures, and our specific primers that we've generated that are going to link up and only bind to segments of our gene of interest. So here come our specific primers. So our specific primers anneal to the two separate strands of DNA. And again, because these are specific to our gene of interest, they don't randomly bind to other portions of DNA floating around. We're selecting for our gene of interest. So once this reaction process has reached the correct temperature, DNA polymerase will come in, and DNA polymerase is going to generate a new strand of DNA that begins where our forward primer starts and ends where our reverse primer starts. So this is where the product size or the product length that we've previously described comes into play. The product that is generated during this process should match the product that we've previously researched while designing our primers. And when we get to testing these on a gel, I will describe and show you why it's important to know the supposed size of your primers. So this was one step, right? This is one cycle of heat and cooling down. So we've cooled back down, and now our complementary strands have found each other again. So we're going to turn the heat on again, right? So now we have here, we're going to heat these guys up again. And now, instead of just our two parent strands, if you want to call them that, we now have our two parent strands and the products of our first cycle of PCR. So, you probably guessed it. Now that we've heated our samples up, our specific primers again come in. They find their complementary sequences from our gene of interest, as well as our newly created DNA strands that are complementary to our gene of interest, and the specific length of our desired product. DNA polymerase comes in again and amplifies again. So in just 2 turns, we've gone from 1 strand of DNA to 4 strands of DNA, right? And I described this earlier. So when we reverse transcribed our mRNA, that occurs in a 1-to-1 ratio when we generate our cDNA. So although this picture looks like we started with 1 copy of cDNA, right, from our 1 copy of mRNA, this is occurring on an order of magnitude reflective of however many transcripts of our mRNA that was present in our original sample. So if we started with 1, there would be 4 right now. If we started with 4, I think we would have 16 right now. So you can see the exponential growth of the total number of our DNA strands and the amplification of our gene of interest. So the animations for this are a little bit out of order because it's hectic to try and get this to work in PowerPoint. But so let's pretend we've fast forwarded a few more cycles, and this is really just to kind of try and represent just how many of these new DNA strands are being generated, right? And again, because we're using primers for our specific gene of interest, this is only occurring in and for our gene of interest. And if you paid attention during our lectures in lab, you'll technically know that I'm telling a little bit of a lie here, because while we're amplifying our gene of interest, in the background we're technically also amplifying one housekeeping gene that we're going to use for a control later, which I'll describe a little bit on the next slide. So after this occurs, we have a smorgasbord of DNA, and now the next step is to see if we've generated A, the correct DNA, and then B, see how the different types of treatments have affected the essentially the ratio of DNA that's produced, right? So remember, the ratio of DNA produced is completely reflective of how many mRNA transcripts there were in the original sample. All right, so now we're on to our last week of our PCR section. So in our last week, we're going to be performing gel electrophoresis on our DNA samples that we generated in the previous week. So these are meant to represent gels for gel electrophoresis again, and then these are wells in our gels. So in each well, a different treatment group is going to go. So over here on our left, we have our ladder control. So this is literally a solution of different chunks of DNA, basically. So we place chunks of DNA that are 1,000 base pairs long, 500 base pairs long, 250 base pairs long. We essentially place all these known lengths of DNA into this well here, and when we run our gel electrophoresis, our control ladder basically will run down the left side of our gel here. And this allows us to figure out how long or what size the DNA products we've amplified in the previous week are. Right? So we, again, to emphasize, we run known lengths of DNA in this lane so that we can compare them to the products we produced in our PCR. So let's pretend the treatment group sample we've placed here is our control. Let's pretend this is a high dose treated, so this group is treated with a high dose of toxin, and this group is treated with a low dose of toxin. And then in our last lane here, when we get to it, I'll point out why this is also important. So imagine technically when we run these experiments, all these samples will be running in unison, and there will also be a loading die in here to help us track how far our bands have run. But for the sake of animation and explanation purposes, our lanes are going to run individually here. So let's say our control sample runs down to 125, right? And so our band intensity looks a little bit like this. So this is our control group. That's what this represents the native quantity of mRNA that's expressed for our gene of interest, right? So how about this guy? So our high dose toxin treated group produces a band that looks slightly lighter than our control group, right? So this can mean a couple of things, right? So our first assumption is going to be that a lighter band means there was less mRNA being transcribed, potentially in response to our high dose of toxin, or it could be something else that I'll comment on later. And then, so our low dose toxin treated group here, for some reason, the low dose of our toxin actually increased the transcription of mRNA for our gene of interest, right? And so that's based on, again, the intensity and literally the pixel intensity. You guys will measure the brightness of each of these bands. So a bright band means there was more mRNA transcribed in that sample, and a lighter band, or a less intense band, would mean there's less mRNA transcribed, right? So in our last group here, this is going to emphasize why we run a ladder of known lengths of DNA chunks on the side of our gels. So if we ran a sample that produced a band way up here by 500, we should know, again, based on the primer sequences that we generated, that those primer sequences will produce a product that's supposed to be 125 base pairs long. So, over here, this can indicate a number of things, but the important thing here would be that the producing a band up here near 500 means that this is not our desired product, likely not our desired product, and essentially means that our PCR failed, again, potentially for a number of reasons. So there's one other instance that hopefully is obvious to everyone that may produce a lower amount of mRNA for our gene of interest, right? So this could be in response to the drug that we treated them with. Our drug could literally just be reducing the total amount of mRNA transcribed for a gene of interest, or there's the possibility that our cells are actually dying, and so as cells die, they likely aren't producing as much mRNA. So maybe this isn't necessarily a drug effect so much as our cells are literally dying from our treatment group. So the way we can control for this, I technically gave it away earlier when I discussed that while we amplify our gene of interest in PCR, we're also amplifying a housekeeping gene, right? So technically when we run our samples, we're going to see something more like this, right? So 3 treatment groups again. So again, control, high dose, low dose, and then this guy over here that I'll explain when we get to him. So the idea behind choosing a housekeeping gene is that the gene, the transcript, mRNA transcript quantity for our gene of interest typically doesn't go up or down very much. It's supposed to be a relatively steady state gene so that insults like these toxins don't change it very much, or if the total transcription of our housekeeping gene is changing, it reflects it can reflect essentially the health of the cell, right? So let's pretend for these three samples that we have nice happy cells that are producing normal amounts of mRNA for critical genes. So these could be genes that are involved in energy production or protein degradation. They're called housekeeping genes because they literally upkeep their house, which is within the cell, so they keep it going and as normal as possible. So if you look here, we have three identical sized bands. So this would mean that despite the treatment group or the dose of the toxin our samples received, our housekeeping gene transcription amount is the same. So in a control, this is the quantity of mRNA that's being transcribed. In our high dose toxin treated group, this is the amount of housekeeping mRNA that's being transcribed, and then the same for our low dose toxin group, right? And so when you guys go to quantify this, you'll literally be comparing the pixel intensity from the band of our housekeeping gene, the band from our gene of interest to the band from our housekeeping gene. So I have some fictitious made up pixel intensities here. So we're going to say that this pixel intensity is 2, and then this pixel intensity is 4, right? So this loosely means that there's half as much mRNA being transcribed for our gene of interest as our housekeeping gene. And I say that because it's not completely accurate, but for the purposes of our lab, the ratio of gene of interest to housekeeping gene is what's important, right? So then in our high dose treated group, say this is a pixel intensity of 1 and our housekeeping gene is still 4. So we can say that this is, 1/4. The ratio of our gene of interest to our housekeeping gene is 1/4. But between the two, because our housekeeping gene is identical, there's half as much mRNA of our gene of interest transcribed in our high dose treated group. And then in our low dose treated group, we have a really, really intense band for our gene of interest. So let's say now that this ratio is 4:4, so it's technically a ratio of 1, right? So the comparison between our gene of interest and our housekeeping gene allows us to more accurately compare the total mRNA transcription going on in our gene of interest. And then this guy right here on the end, so if you look closely, the pixel intensity of this gene of interest is really, really low, even compared to our high dose toxin. So maybe this is what we see, right? We see a reduced pixel intensity or a reduced quantity of mRNA for our housekeeping gene. So if we measure this, maybe this ratio comes out to be 0.2 to 1, right? So even still, with this being so drastically different from our other treatment groups, we can now establish that the ratio of our gene of interest to our housekeeping gene is 0.2, which will allow us to compare it to other treatment groups.